Simon Ferrier is works at CSIRO at Canberra. He's been involved in biodiversity modeling for a long time now. He's been one of the early people doing sophisticated species distribution modeling, and is particularly excited and strong in trying to think about not just single species, but whole communities of species and how to model them in space. So he's a real leader in that, and also very active in the science policy interface, and hopefully be able to get that going. So particular thanks from Simon, who came all the way from Australia. He's been with us for 10 days, 2 weeks, so our first sabbatical visitor to the program. Fire away, Simon. Thanks.
Thanks very much, Walter. As this symposium goes on, it's getting harder and harder to say something that hasn't already been said. And for those of you who were here yesterday, you'll notice that this diagram looks very similar to one that Ben Collin put up yesterday. So there's some nice convergent evolution going on in our thinking. I'm going to be talking about three biodiversity challenges, assessment challenges, and Ben did touch on these yesterday. The first of these is to do with observing change in biodiversity, past to present. The second challenge is about trying to project what is going to happen to biodiversity or could happen to biodiversity in the future, in other words, scenario analysis. And the third challenge is the one of then using modelling of what we think might happen to that future trajectory of biodiversity if we were to implement different types of policies or management actions as a basis for prioritising and making decisions in relation to those actions. Okay, now when we look at that state of biodiversity y-axis there, we can imagine that can be assessed at many different spatial scales. And I should say right up front, and this again gets back to something that was talked about yesterday, and that is that most of my previous experience before moving to CSIRO 4 years ago was actually within a state government environmental agency. And so most of the work that I was involved with was actually at the state or regional or landscape scale, and it was all about planning and decision making. So just to give you a bit of that background on where I've come from. I've actually come from a background where I've been involved in real world decision making at the landscape scale or at the regional scale. And so my interest in thinking at whole continent scale or at global scales is relatively recent. Okay, so this can happen at different spatial scales. We can also think of state of biodiversity as being assessed in relation to different dimensions and levels of biodiversity. And so there I've just got an adaptation of the old Reed-Noss perspective on the dimensions and levels of biodiversity. And when we look at that and we think back to Woody's talk, then it becomes obvious that there are some levels and dimensions of biodiversity that are a lot easier to assess remotely than others. So things like ecosystem structure and function, people are doing a reasonably good job of assessing those remotely. But when we come to composition, other than some of those things that Woody mentioned, like the floristic composition of canopy trees, for example, that you can now assess to some extent with hyperspectral imagery, most of the composition of biodiversity at the species and genetic level quite obviously we're not in a position to assess that remotely. So this presents some very real challenges. And so I'm going to be focusing most of my talk on this compositional dimension of biodiversity and on the challenges of assessing that at the global scale. And if we look at how people have approached this problem before, there are two commonly employed approaches in global assessments of compositional biodiversity, one of which is to look at or to work with distributions, for example, range maps of species in better known biological groups, such as the vertebrates, and Walter talked a lot about this yesterday. And another fairly commonly used approach is one where people focus on mapped ecosystem types or biomes or ecoregions and essentially use those as coarse filter surrogates for biodiversity composition, and use approaches like species area relationships to infer what's happening in terms of the retention of compositional diversity. But there's been a lot of work done over the years, and I was involved a lot with this sort of about 20 years ago, of evaluating how well some of these surrogate approaches perform for the unsurveyed elements of biodiversity, or in other words, the other 99% of biodiversity. So all of the invertebrates, the lower and higher plants, the microbes and so on. How well are these catered for by these surrogate approaches? And I think, to summarise the story there, the results of a lot of those assessments have been quite mixed. So there's some real issues with using things like vertebrate species ranges and broadly mapped ecosystem types and ecoregions and so on as surrogates for the rest of biodiversity, or the other 99%. But then, on the other hand, we know that for a lot of these neglected groups, there's a surprising amount of information out there. And this is just one example of a group that we're just starting to do some modelling with at the moment, globally, the Diptera, or true flies. And when you look at that group, there are over 3 million records for almost 120,000 species of Diptera within GBIF. There's also now quite a superb high-level phylogeny of the whole Diptera. So this is quite an amazing amount of information on a group like that. Okay, but as you would have seen from that map on the left there, the geographic coverage of those records is patchy, and there are big gaps. So we don't have geographically complete data, hence the idea of integrating in situ or field surveyed biological data with remotely mapped environmental variables. And Woody's already talked about this quite a bit, and this can be used at the most basic level just to model spatial pattern in the distribution of biodiversity. If we get a little bit more sophisticated, we can bring in remotely observed change in ecosystem state or intactness to infer changes in the status of biodiversity. And then in turn, we can get down here to bringing in projected pressures and responses, things like climate change, land use change, to project biodiversity into the future. So Woody's talked a fair bit about this, so I won't dwell on that too long, but just jump straight to this slide, which is just simply to point out that as soon as you mention linking biological data and remotely mapped environmental data, a lot of people immediately assume that you're talking about species distribution modelling or niche modelling. That's certainly one very important strategy for linking these sources of information, and I've spent a lot of my career deriving species distribution models. I modelled my first species in 1982 and then modelled many thousands of species in the 15 or 20 years following that. But species distribution modelling is just one way of integrating these sources of information. And there are a lot of options there that I won't go into. If you're interested in learning more about that, then have a look at that review paper that I wrote with Antoine Guissan a few years ago in Journal of Applied Ecology. But I just wanted to focus on the two ends of the spectrum there. One end is the individual species distribution modelling that we're all very familiar with. But down at the other end of the spectrum here is another approach which we're now using quite heavily, which is what we call macroecological modelling of collective biodiversity properties. And so this is modelling things like richness and compositional turnover across space. And I'll be talking about this in a little bit more detail soon, but the point I wanted to make here was that we've got all of these different modelling strategies, and it's really horses for courses. The appropriateness of different strategies depends very much on the question of interest and the nature of the biological group that you're working with and how much data you've got within that group. So if you're working with a group like birds, where society places value on each and every bird species and they all have names that people are familiar with and so on, and you've got a reasonable number of records per species, then it makes sense to be modelling individual species distributions. But if you get down to this end of the spectrum here, where the group you're working on, people aren't really valuing the individual species within it, but we do value the overall diversity of that group, and also we've got a huge number of species in that group, a lot of them with very few records and probably a lot that don't have any records at all and we don't even know that those species exist, then this other kind of strategy might make a lot more sense in that situation. So what do I mean by modelling collective properties of biodiversity? This is probably fairly obvious, but the one that has been done a lot more is where people model local richness or alpha diversity, so something like the species diversity at a location as a function of the environment of that location, the biogeographic history, human disturbance, and so on. Or it becomes a lot more interesting when we start to also bring in consideration of the compositional dissimilarity between locations, or in other words, beta or beta diversity, and trying to model that as a function of differences in the abiotic environment between those locations, the level of biogeographic isolation, and those sorts of things. Now, with all of this modelling, we can be talking about just simple measures of species richness or compositional dissimilarity in terms of species overlap, or we can get a little bit more sophisticated and bring in considerations of phylogeny. So all of these modelling approaches have phylogenetic versions of them. Now, just to bring this to life a bit, here's an example of work that we've done within the whole Australian continent, going back a couple of years now, where here we're focusing primarily on modelling those patterns of beta diversity or patterns of compositional turnover across the Australian continent. And this is a rather different approach to the individual species modelling approach, in that this is a model, in this case here, and sorry if you can't see those fitted functions there, they're obviously very faint on the screen. But the important point is that this is a model that's working with compositional turnover as a collective property of this particular group, which for the example I've got here is land snails, and we've modelled lots and lots of different biological groups within Australia. But this is a model that's fitted to a whole lot of data for almost 3,000 species of land snails. So we're looking at all the data for land snails together within a single model, relating that to environmental variables to do with climate, terrains, soils, also bringing consideration of geographic isolation into the modelling. And this is all used to then say something about the spatial pattern in the distribution of biodiversity for that particular group, in this case the land snails across the Australian continent. And so that then in turn is used, if we do that for a lot of different biological groups, that then provides the basis for doing all sorts of assessments that are built on top of that. This is just one example of work in the last couple of years where we've done climate change impact analyses based on those sorts of models to help inform national scale policy development and planning. Okay, but what's all this got to do with the global scale? Well, we first had to go at applying these kinds of approaches at a global scale in the lead up to the 5th World Parks Congress in 2003, and we were essentially complementing some of that work that got reported yesterday, work done by Tom Brooks and others, by showing as a proof of concept how you could apply our sorts of approaches to best available biological data sets drawn from representative combinations of biogeographic realm and biome and linked to best available environmental layers at that point in time. So this was used as a basis for assessing the representativeness of the global protected area network. In the years since then, a lot has changed and has moved forward. So we're now in a position where I think there are a whole lot of new opportunities opened up for the application of these approaches, because we've got things like GBIF and related initiatives have really moved forwards in leaps and bounds over the past almost 10 years. We've got all the work that Woody was talking about with GEO and GEOS in terms of Earth observation. There's a whole lot of work happening with global scenario analyses with things like the image model, and we've also got these new policy initiatives relating to the Convention on Biological Diversity and the new Aichi targets, and also the emergence of IPBES. So there's suddenly this new window of opportunity to apply some of these techniques in a more rigorous way. And so we, about a year ago, had another go at a proof of concept analysis that made use of some of these approaches using the improved information that's now available. And this is very much, as I said, proof of concept. It's just to give people the flavour of what might be possible, hoping to then leverage the sorts of resources that are needed to really roll this out properly. So in this case, and I won't have time to go into it in great detail, we're bringing together the best available data on all the fern species for the world, over 1.3 million records for about 10,000 species of ferns from GBIF, combining that with best available environmental data, land use change layers at a fairly coarse resolution from the image model, climate change projections, bringing all this together with modelling approaches that I won't go into in detail now, but happy to talk about them with anyone later, bringing all that together to then say something about what we think is happening to the retention of compositional diversity within this group. Okay, that then allows us to say something about the expected changes at different spatial resolutions. I won't try to explain that one in detail, but the important point is that because of the way we do this and the way we're considering alpha, beta and gamma diversity, we can in fact aggregate up these results to different spatial scales. So we can say something about what we think is happening, for example, with an individual ecoregion, with the whole biogeographic realm, or even with the whole planet. Okay, just to very quickly run through what are some of the current directions in the application of these kinds of approaches. And some of this has already been touched on by others, so I won't dwell on parts of it. But in terms of the types of biological data that we can use in this kind of modelling, clearly, citizen science is changing the whole ball game there. But the other big change now is environmental metagenomics, and we're working a lot now with people who are doing metagenomics, not just for microbes, but also now for things like arthropods. And this is a way of going out and getting these big grab samples of the environment using fast throughput sequencing to get a complete profile of the biodiversity within those samples. So this is a big step forward. Woody's done a superb job of covering the remote sensing end of things, so I won't dwell on this, but obviously hyperspectral LiDAR and radar are game changers. And there's a lot of very exciting work happening with the derivation of new fine-scaled abiotic surfaces, climate and soils, etc. I've just got one example there drawn from work of some of my CSIRO colleagues in Canberra, who are all linked into this big new global initiative. In fact, CSIRO is one of the leaders of the Global Soil Map initiative, which is trying to transform the way that soil attributes are mapped globally. And so they have been trialling approaches that are going to be rolled out globally for mapping soil attributes from remote sensing integrated with a whole lot of other sources of information and various forms of modelling. So that's a very exciting step forwards. In terms of directions in modelling spatial pattern and the distribution of biodiversity, we've been doing a lot of work on smarter ways of incorporating phylogeny, accounting for biogeographic history, and also dealing with the ever-present issues of bias and incompleteness in biological sampling. We've also been doing a lot of work on getting smarter about how we do biodiversity projections under global change scenarios, incorporating ecological mechanisms and processes into our models, and also starting to get serious about accounting for the capacity for evolutionary adaptation in these projections. And if I can just flash up a couple of examples of that, this is work led by one of my postdocs from the past 3 years, where this is Karel Makani, he's been extending the sorts of macroecological models that we've been used to project future changes in biodiversity. He's been extending those models to better consider aspects of ecological processes. So in this case, community assembly rules and dispersal are actually incorporated into that modelling. And in terms of, I mentioned capacity for evolutionary adaptation, this is work that is just starting up, and this is a very big project, a joint project between CSIRO, Melbourne University and Monash University, which is looking at the genomics of climate adaptation. And that's actually using a number of model groups of organisms. Drosophila is one, eucalyptus is another, and there are a few more, but essentially they're working on getting a lot smarter about being able to predict, estimate or infer the capacity for evolutionary adaptation across different sets of organisms. And so the Drosophila are quite an extraordinary group to work with in this regard. I think Woody was talking about the whole enchilada before. The Drosophila are definitely the whole enchilada. They have, within Australia, for the Drosophila group, they know absolutely everything you would ever want to know about this group in terms of what it does or what climate change does to the group, right through to running experimental evolution under climate change. So it's quite amazing. And this is going to provide the most superb test bed for then evaluating all sorts of other approaches that people have used to model biological responses to climate change. Okay, then finally, this box out to the side here, which I haven't mentioned so far, which is a critically important one, and that is the evaluation and calibration of some of these sort of remotely derived models of what's happening to biodiversity, and evaluating and calibrating those using long-term field monitoring plots, ecological plots and biodiversity plots, a really crucial part of the whole story. And then there's also potential here to make use of things like paleoecological data to evaluate the performance of these models. Just to touch on that very quickly, because I know Walter wants to catch up on time, but this is just an example of where within Australia we're putting a lot of work into sort of trying to link these two communities of practice, the more sort of remotely derived model-based approaches to biodiversity assessment, and then the community of practice that is more geared towards going out and repeatedly surveying biodiversity at field plots over time. And so we're really interested in better ways of actually integrating these two approaches and getting the right feedback loops going on in there in terms of calibration and evaluation. And then with the paleoecological example, this is a paper that's just out that's based on ongoing work that I'm doing with Jack Williams and Jessica Blois from University of Wisconsin, where they have this most incredible fossil pollen data set from the last 20,000 years that is essentially allowing us to look at how well some of our predictions of biodiversity change over time based on these macroecological models hold up when you look at what's actually happened in terms of the pollen record. So that's a whole story in itself. Just the very last slide, Walter, and I think Woody said that the key word was integration, and here it is. I couldn't agree more. The whole challenge that we've got is one of integration, and I think it's integration across lots of different dimensions. So just to run through them very quickly, the challenges I see are integrating change observation, scenario analysis and conservation planning, which at the moment sort of tend to operate in parallel universes, different communities of practice. Integrating across scales, local, regional and global, integrating the compositional, structural and functional perspectives on biodiversity, obviously integrating remote sensing and in situ observations, and also, really importantly, given what I've just talked about, the need to integrate species-level approaches and macroecological modelling approaches to this integration of remote sensing and in situ observations. So both are really important. They're highly complementary, and we've got to get smarter about how we use them in conjunction with each other. Thank you.
Thanks a lot, Simon. Thanks for keeping on time. Gives us time for 1 or 2 questions. But in a minute or so, you can already come up, Matt, and get started. But questions? Catherine.
Simon, maybe too specific, but you had, when you were modeling compositional similarity as a function of the biocom variables, so then you sort of have these arrows sort of suggesting that you had future climate and future land use somehow integrated in them to come up with some sort of assessment of what we might lose. Can you tell me how those things were integrated?
If I had 10 minutes now, which I don't think Walter will give me. So if we can talk afterwards, that takes quite a while.